Chapter 2 Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using the criteria that were scientific, but born out of need to use organisms for own use, for food, shelter, clothing. Aristotle was the earliest to attempt a more scientific basis of classification. He used simple morphological characters to classify plants into trees, shrubs, herbs. He also divided animals into two groups, which had a red blood and those did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdom was developed, that included all the plants and animals respectively. This system did not distinguish between the eukaryotic and prokaryotic, unicellular and multicellular organisms, photosynthetic and non-photosynthetic, that is, green algae and fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand. But a large number of organisms did not fall into either category, that is, either in plants or either in animals. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships. Classification systems for the living organisms have hence undergone several changes over time, though the plant and animal kingdoms have been constant under all the different systems. The understanding of what groups organisms be included under these kingdoms have been changing. The number and the nature of their kingdoms have also been understood differently by different scientists over time. Characteristic of five kingdoms. Cell type of Monera is prokaryotic, Protista eukaryotic, fungi eukaryotic, Plantae eukaryotic, Animalia eukaryotic. That is, only prokaryotic cell type is of Monera. Cell wall. In Monera it is non-cellulosic, in Protista it is present in the sum, and in fungi it is chitinous. It is fungi. In the fungi it is made up of chitin. In Plantae cellulosic, in animals it is absent. Nuclear membrane. In Monera, as it is prokaryotic, nuclear membrane will be absent. In Protista, nucleus is present, therefore nuclear membrane is present. Fungi, it is present. Plantae, it is present. Animalia, it is present. Body organization. Monera is the first kingdom, therefore the simplest, cellular. In the Protista, cellular. In fungi, multicellular or loose tissue. Plantae, tissue, organ. In Animalia, tissue, organ and organ system. Mode of nutrition of Monera: autotrophic, that is chemosynthetic and photosynthetic, and heterotrophic. In heterotrophic it might be saprophytic or parasitic. In Protista it is autotrophic, photosynthetic and heterotrophic. Fungi heterotrophic, either saprophytic or parasitic. In Plantae autotrophic, photosynthetic. Animalia heterotrophic, holozoic nutrition or saprophytic nutrition. R. H. Whitaker in 1969 proposed five kingdom system of classification. The kingdoms defined by him were named as Monera, Protista, fungi, Plantae and Animalia. The main criteria for the classification used by him include cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationships. Table 2.1 gives a comparative account of different characteristics of five kingdoms. The three-domain system has also been proposed that divides Kingdom Monera into two domains. One having nucleus, that is prokarya, and another one as eukarya. Eukarya have nucleus, leaving the remaining eukaryotic kingdoms in all the third domain. That is, under the eukarya there is Plantae, Animalia, fungi and Protista. Under the prokaryotic, it includes only Protista, that is, 6 kingdom of classification. You will learn more about this system in detail in higher classes. 3 domains. Kingdom Monera into 2 types. And remaining all 4 kingdoms into one domain, that is Eukarya. Therefore, these 4 plus 1 plus 1 total 6 kingdom of classification. Let us look at the 5 kingdom classification to understand the issues and consideration that influence the classification system. Earlier classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms and angiosperms under the plant. Currently, only mosses, ferns, gymnosperms and angiosperms are plants. Bacteria, that is, they have separate kingdom named Monera. Blue-green algae, that is cyanobacteria, is also a bacteria. Fungi has its own kingdom called kingdom fungi. So the bacteria and blue-green algae are monarians and fungi has separate kingdom, and all others are included in plants which were earlier placed together. The character that unified this whole kingdom was that all the organisms, that is bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms and angiosperms, had cell wall in their cells. This placed together groups which widely differed in other characters. It brought together the prokaryotic bacteria. Bacteria are prokaryotic. That's why they are placed in Monera. And the blue-green algae, that is cyanobacteria with other groups which were eukaryotic, that is, they placed the prokaryotic bacteria and the blue-green algae with other groups which were eukaryotic, as both the prokaryotic bacteria and the cyanobacteria are prokaryotic. So this was wrong. It also grouped together the unicellular organisms and multicellular organisms. For example, Chlamydomonas and Spirogyra, which is currently a fungus, and the Chlamydomonas is a protistean, were placed together under algae. Sorry, the Spirogyra is algae, but the Chlamydomonas is a protist. The classification did not differentiate between the heterotrophic group of fungi and the autotrophic green plants, though they showed a characteristic difference in their wall composition. The fungi had chitin in their cell walls, while the green plants had a cellulose in their cell wall. While such characters were considered, the fungi were placed in a separate kingdom, that is, kingdom fungi. All prokaryotic organisms were grouped together under the kingdom Monera, and the unicellular eukaryotic organisms were placed under kingdom Protista. The kingdom Protista had brought together Chlamydomonas, Chlorella, earlier placed in algae with plants and both having cell walls, with Paramecium and Amoeba, which were earlier placed in kingdom Animalia due to lack of cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for the classification changed. This kind of changes will take place in the future too, depending upon the improvements in our understanding of characteristics. Evolutionary relationships. Over time, an attempt has been made to evolve the classification systems which reflects not only morphological, physiological and reproductive similarities, but also phylogenetic similarities. It is based on evolutionary relationships. In this chapter, we will study the characteristics of kingdom Monera, Protista and fungi of the Whittaker system of classification. Basically, we are going to study the Whittaker system of classification. The kingdoms Plantae and Animalia, commonly referred as plant and animal kingdom respectively, will be dealt separately in chapters 3 and 4. 2.1 Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans where very other forms can survive. Many of them live in or out of organisms as parasites. Bacteria are grouped under four categories based on their shape: the spherical coccus, the rod-shaped bacillus, spiral-shaped spirillum, comma-shaped vibrio. Though the bacterial structure is very simple, but they are very complex in behavior. Compared to many other organisms, bacteria as a group show most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substances. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of the bacteria are heterotrophic. They depend on other organisms or dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats. NEET 2020 2017, such as extreme salty areas, that is halophiles, hot springs, thermoacidophiles, and marshy areas such as methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of the several ruminants, such as cows and buffaloes, and they are responsible for the production of methane, that is biogas, from the dung of these animals. Eubacteria. There are thousands of different bacteria. EU true, that is true bacteria. They are characterized by the presence of rigid cell wall and, if motile, flagellum. The cyanobacteria, also referred as blue-green algae, has chlorophyll A, similar to the green plants, and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial, or filamentous. Freshwater, marine, or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts, for example, nostoc and Anabaena. The chemosynthetic autotrophic bacteria oxidizes various inorganic substances such as nitrates, nitrites, ammonia, and they release energy for their ATP productions. They play a very great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are the most abundant in nature. The majority are important decomposers. Many of them have significant impact on human affairs. They are responsible for the curd making from the milk. Production of antibiotics, fixing nitrogen in legume roots. Some are pathogens causing damage to the human beings, crops, farm, and animals and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by the bacteria. The bacteria reproduce mainly by fission, sometimes under unfavorable conditions. They produce spores. Unfavorable conditions, they produce spores. They are also reproduced by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to another. Mycoplasma are the organisms that completely lack cell wall. They are the smallest living things or living cells known and can survive without oxygen. Many mycoplasma are pathogenic to plants and animals.